Welcome to the Diabetes Bio Podcast, a podcast for the Diabetes Journal, the flagship research publication of the American Diabetes Association. Each month, we interview authors from our feature articles, papers of the month, or other articles published in that month's print issue of the Diabetes Journal. We also have a Sweet Talk segment where we get a little creative in our scientists and topic interviewees. And we have another great episode for you today. In fact, it is our 24th episode. We have been doing this for 2 full years. What do you think?
Woo-woo.
Yeah, it feels like a rhythm now.
Yeah,
Yep.
it does.
And I think we might actually add a couple more in there with the live episodes, right? Or no?
That's true. That's
Oh,
true.
that's right.
Yeah.
So
That's right.
26, I guess.
Yeah. So I think today we will continue our tradition of talking about our favorite interviews of the year, but I also wanted to ask you what is the thing that has surprised you about doing this podcast? Has there been anything that you didn't expect in this process?
I think I've enjoyed it more than I thought I would.
Yeah.
It's always fun to talk about science. And we get to talk to people who I think I may have mentioned this before. We never really may have interacted with just because it's a little different type of diabetes research outside of our wheelhouse. And it's been really nice to have those conversations. And I've enjoyed it more than I thought I would.
Yeah. What about you, Kevin?
I second that for sure. I mean, the people and the conversations, learning so much about the science, but also about the individuals and their career paths and trajectories and what took them to where they are today. You know, these are things that are not well known or appreciated for people in the field. I think another aspect is really cool getting the feedback, going to meetings, going to universities, giving lectures, or just running into people randomly, and they come up to you and say, "Hey, I've listened to your podcast. I really enjoyed it. I get this or that out of the conversation that you've had with so-and-so or different individuals." And I think that type of where we're touching people in the field really strikes a chord, and something that I didn't appreciate when we started these episodes.
All right, Dar.
Yeah, something you said, Kevin, really made me think about I think we've seen more and more investigators. In fact, one of my favorite interviews of the year was Stephen Kahn. But more and more folks, Tony Ferrante is another one, if you haven't seen him present lately, that are making a point of talking about the importance of communicating your science to everybody, right? Not just each
Absolutely.
other. One of the things that comes through, I think, in a lot of these interviews, and I think today is a really good example of that, is that people really enjoy their science, and it comes through in the way that they talk about it, right? So I think the lessons for the younger folks, in addition to listening to people's career journeys, which are a lot of times, maybe not the traditional path and still led them to someplace really cool. It's not always a step 1 leads to step 2 leads to step 3 kind of thing. And people still have really good success. And so those are 2 things I think junior folks can take away from this, right? Like, hold on to your passion for science no matter what. And that career journeys aren't always linear. And so, yeah, hopefully that has some impact on the junior folks to keep them going in these sometimes uncertain times.
Yeah, the non-circuitous route was, it brought me back to, it's not a conversation we had this year, but we talked to Giles Yeo last year in November, which was Diabetes Awareness Month and Diabetes Awareness Day, or World Diabetes Awareness Day. And he talked about his path, right? And he's somebody I definitely look up to as a great science communicator. And if you listen to his story during that episode, really his spotlight in going out and doing this science communication was because he was taking a step back away from his research, his primary job of what he was doing in the lab, and exploring other opportunities. And we might not have him as that type of a science communicator had he not thought about these other opportunities. And of course, by doing this, he also reinvested back into his research. And so he's very active in what he's doing today, and very productive, and bringing a lot of insight into metabolic regulation through central circuits. So I just found that, kind of story, as well as many of the other stories that we've heard from these investigators, and like you talk about, not a linear path. It's very interesting to hear those stories and where their careers have gone. And you can definitely get from it that we were talking to these different award winners that they never really expected to be where they are today. It's just their path went this direction. And of course, they are very bright individuals and can communicate their science and tackling tough questions. They ended up where they are today.
Yeah. So what were your favorite interviews this year?
I'll say that I really enjoyed having Dan Drucker and John Campbell on.
Yeah, that was a good one.
It's first of all, they're great. They're both great to talk to.
Yeah.
Independently. And also there's something cool about, seeing the mentor mentee, even after, they're no longer, I guess, your official mentor anymore. But there's always sort of that that protective or proud parent sort of thing that comes through.
Yeah. Yeah, for sure. Kevin, what about you?
Well, I thought about this, and looking back on the year, I've just been so impressed with the content that we've been able to put together. We've talked to real luminaries, real leaders in the field of diabetes and metabolic research. We've talked about, or we've chatted with previous Banting Medal Award winners, Outstanding Scientific Achievement Award winners. Darlene already mentioned Stephen Kahn. Kirk mentioned Dan Drucker. We've also chatted with Jeffrey Flyer, Philip Scher, Joel Elmquist, Jens Yul Holst, Barbara Kahn, Shingo Kajimura. We've chatted with Pathways to Diabetes Award recipients, including Alexander Nektow and Classics in Diabetes series, Brett Goodpaster. I mean, it's just been so impressive, I think, this last year, and it's really difficult to choose between any of those individual episodes. I really recommend the listeners to check them out. But if I had to choose just one episode, I think the one that I would go for is our October episode on the childhood obesity and diabetes. And that was with Bethany Cartwright from UT Southwestern, Kristen Nadeau from the University of Colorado, Anschutz Medical Campus in Denver. The conversation covered many important issues in the biology behind obesity and diabetes, the treatments that are available, weight stigma and bias. And I think a really special thing of that episode was to bring the kids in and ask questions that were personal to them. We had kids that were ages from 4 to, I think it was up to 16. And I know Darlene and Kirk are going to remember some of these about, questions as basic as why sugar is bad for you, the serious side effects of long-term uncontrolled diabetes, like amputations, and an interest in something that, we might not necessarily think about as we're thinking about in the aspect of treating diabetes and obesity, but the interest of wanting to gain weight in a healthy way for kids to play sports and different types of activities. And so I think it was really cool to have the kids and their curiosity, really their honesty to the conversation. It's an episode I'm really proud of, and I think it touched a lot of people. Looking at the listeners and the downloads that we've had, it's only been out for a couple months, and it's already topping one of our top downloads for the year. So I really highly recommend checking out that episode.
It was a little bit of genius, right? Because it's a lot of those kids' family members that are listening. Grandma and grandpa.
That's right. That's right.
Suddenly our numbers have picked up. It was a really good episode, though, and kudos to Kevin for the creative thought process in generating that idea. That was really
Yeah,
fun.
he gets credit for that idea. That was a good one.
For sure. I mentioned Stephen Kahn's interview already. I did really enjoy that one. I mean, he's a delightful person, but I also like today's, and I don't want to go too much into detail, but one of the reasons that I like today is both because both of these, well, all of these authors, I should say, have a real infectious enthusiasm for the work they're doing, which I loved. But then also the ability of Neelanjan Mukherjee and Holger Russ, their ability to explain their genetic, their heavily genetic work to the physiologists, I thought it was really good. And I really enjoyed that.
And it was so cool that we had this weird overlap between their trainees that preceded even the collaborative effort of the groups, right?
Yeah.
It was
Yeah.
just this weird coincidence in science that probably happens more than we track.
Yeah. Yeah. We can't underestimate serendipity and the influence of our scientific projects.
And
And
your
then
network.
Bryan Bergman is just he's so enthusiastic about his science, and he does a really good job explaining it. So yeah, so that was my favorite.
So encourage the listeners to listen to this episode.
Yeah, exactly. We got a good episode. So let's talk
Yeah.
about it. Kirk, why don't you tell us a little bit more about the titles and authors?
All right. Darlene has buried the lead on this one.
I
But
did. Spoiler alert.
with December, we're going to start with the paper of the month. Proteogenomic discovery of novel open reading frames with HLA immune presentation on human beta cells. This was a joint venture, as we mentioned, between the labs of Neelanjan Mukherjee and Holger Russ. We also had the opportunity to discuss one of December's feature articles titled Combined Weight Loss and Exercise Training Alters Skeletal Muscle Subcellular Lipid Localization and Intramuscular Adipose Tissue Cellular Composition. In this segment, we were joined by a corresponding author, Bryan Bergman, who is a professor in the Division of Endocrinology, Diabetes, and Metabolism at the University of Colorado Anschutz Medical Campus. These were both great discussions and a lot of fun.
So unfortunately, I missed the recording of the Sweet Talk segment due to
How
scheduling.
dare you?
I know. I was very disappointed. Nothing personal to these amazing people, but Kevin, why don't you tell us about it?
Yeah, it's a great one. Today we're talking about something that's really fundamental to the entire field of metabolic research, how we actually measure metabolism. Scientists have been using animal models and several tools like indirect calorimetry to study energy balance. But as science has advanced, so have the methods. We now have better ways to collect that data, more powerful statistical tools and standardization methods to interpret the data, and new discussions about how these approaches translate to humans. And to help us unpack all of this, we're joined by three leaders in metabolic research: Dr. Alexander Banks from Beth Israel Deaconess Medical Center and Harvard Medical School, Dr. Corey Holman from the University of Pennsylvania, and Dr. Louise Lantier from Vanderbilt University. Together, they're discussing how the field is progressing in measuring, analyzing, and interpreting these metabolic data. I think it's a fantastic topic, so stick around and to learn more.
All right, let's dive in.
Today, we will be discussing one of December's feature articles titled Combined Weight Loss and Exercise Training Alters Skeletal Muscle, Subcellular Lipid Localization, and Intramuscular Adipose Tissue Cellular Composition. We are joined by corresponding author, Dr. Bryan Bergman. Dr. Bergman is a professor in the Division of Endocrinology, Diabetes, and Metabolism at the University of Colorado Anschutz Medical Campus. Dr. Bergman, congratulations on your paper, and thank you for being a part of this month's podcast.
Hey, thanks for having me. Excited to be here.
All right, Bryan, if you're a listener of the podcast,
Well,
you know
who
that
isn't? Everybody listens to this thing. So yeah, totally.
well, then you know that we always start with an icebreaker question. So my question for you is, how does your personal life intersect with your work?
Oh, good Lord. How does it not? I
First of all,
personally,
daughter, that's none of our business.
I don't actually draw many distinctions between my personal life and my work life. I don't know if you guys are the same way, but to me, the two are very much intertwined. I mean, I try to live a really clean life and exercise every day because of the work that I do. Because of all the work that I'm doing with intramuscular adipose tissue, I don't eat a lot of red meat anymore. I mean, it's just all these things are changing because of my work. And so there's a tremendous overlap between the two.
Yeah, for sure.
And plus, my wife is an endocrinologist here. We talk about study designs and science until late in the night. And so it's just sort of part of my everyday, honestly.
So, Bryan, before we discuss the article, we always like to give the authors a chance to highlight any other contributions that have been important to the paper, other authors, collaborations, anything like that.
Yeah, well, first is Corinne Zemsky-Berry. She's the first author on this. She did an amazing job with this paper. So I just wanted to give her a shout out. She couldn't be here today. And then just my whole collaborative team. I mean, I have an amazing group of people that I get to work with every day. And I feel very, very fortunate. And of course, this was funded by the ADA many, many years ago. So my apologies, it took so long to get out. But of course, the ADA for funding this work.
Yeah, of course. How does this well, I guess let's take a step back. Let's start with an overview of your research program. And then how does this project kind of fit into, or how did it grow out of that program as a whole?
Yeah, it's a good question. And I'll try to distill this down and not be too long-winded. So the overall end all be all of my work is to try to come up with new therapeutic targets to treat or prevent type 2 diabetes. And my focus has always been on muscle because it's the biggest sink for glucose disposal after you eat a meal. And yet we don't have any targeted therapies really designed to make muscle more insulin sensitive. Metformin has some off-target effects. Of course, weight loss and exercise training are fantastic. And until recently, weight loss was really, really hard. It still is for people, but most people can't stay with kind of a fitness program long enough to make that a good thing for them. And so I've always just been focused on muscle. And coming out of my PhD, where I was doing a lot of exercise physiology, I was really, really excited about this idea of the athlete's paradox that Brett Goodpaster published many, many years ago, where athletes have a bunch of triglyceride in their muscle, yet they're super insulin sensitive, and everybody else with that much triglyceride is very insulin resistant, and what's going on there. And it was a great intersection of everything that I've been studying up to that point in XPhys and my new love for diabetes. And so that's really what started this whole thing going. And up to, I don't know, maybe 2010 or so, a lot of the field were just sort of grind and find biochemistry. You take some muscle, you grind it up, you see what's in there. More is worse is kind of where everything was in terms of lipids and insulin resistance. And then we started realizing, there's all these different molecular species. Let's look at that. And that seemed to answer some of the questions in the field, but it definitely didn't explain a lot of the times where you could have somebody become more insulin sensitive and lipids increase, or lipids decrease and insulin sensitivity didn't change, et cetera. So we just started thinking more and more about how these lipids work, where they're working, and it made us realize that we're really in the caveman era of this work, and we really need to figure out where these lipids were located in the muscle cell to see how it might be related to insulin sensitivity. And that started this whole ball rolling of probably 10 to 12 years of work where we really try to pursue it. And so that's sort of the ideology of this whole thing.
Yeah, that's a great overview.
Awesome. So let's talk a little bit more about IMAT. So your article investigates how combined weight loss and exercise training affects metabolic markers, but specifically, again, this sort of skeletal muscle subcellular lipid localization of the intramuscular adipose tissue, which we will now call IMAT. So maybe talk a little bit more about IMAT and why, maybe expand on why targeting this depot has become an important area of interest in understanding skeletal muscle and whole body insulin sensitivity.
Yeah, great question. I think IMAT is the next big thing. And all of the work that's going on in my lab is really focused on IMAT. And IMAT's funny. To me, it's this therapeutic depot that's been hidden in plain sight for our entire lives. It's in everybody's freezer. It's every time you walk through a grocery store, you see it, but you just don't recognize it, right? So IMAT is the marbling in a steak that is in every one of our muscles. It's the adipose tissue that surrounds all of our muscle fibers. And it was, again, I always follow Brett's work, but Brett first published that this was an important depot in terms of its relationship to insulin sensitivity. And a lot of the work that followed that really validated the fact that every time you looked at it, how much IMAT was in somebody's muscle was directly proportional to how insulin resistant they were. It explained the sarcopenia associated with aging and obesity, explains insulin resistance associated with sex, race, and ethnicity. I mean, every time it was looked at, it was an important player, but nobody really knew if it was truly a causal factor or just related to visceral adipose tissue. And that's really what was driving this relationship. And so one of the things that my lab has been trying to do is figure out, okay, how is IMAT, if it truly is causing problems, how it might be causing problems. And so we've been searching to sample it and target it specifically. And after making a bunch of mistakes with IMAT that we pulled out of muscle biopsies, we finally started to work on sort of a very simplistic way of thinking about it. If this adipose depot is right next to muscle, maybe it's just secreting factors, paracrine signaling, that is causing the muscle to have problems. And so we started taking the IMAT out of these biopsies, culturing it to make conditioned media, and then looking at how it might be impacting insulin sensitivity and sarcopenia risk. And so what we found is that sort of the moral of the story is that IMAT is actually worse than visceral adipose tissue because we've compared them head to head in terms of its ability to cause decreased insulin sensitivity, inflammation, etc. in muscle. So it is this really negative thing. Every way I look at it, it's always this negative impact on muscle that's constantly bathing our muscle with all these factors that are promoting decreased insulin sensitivity. So that's why I think it's important, because if we could antagonize any one of these signals that is promoting decreased insulin sensitivity in muscle, we could have, I think, a huge impact on preventing and treating type 2 diabetes.
Now, to build on that, can you take us maybe through some of your prior work that led you to hypothesize about the subcellular localization of those lipids? And then also the specific lipid species that may affect insulin sensitivity when we're talking about IMAT and skeletal muscle.
Yeah, I draw a distinction between the two. IMAT is adipose tissue outside the muscle. It
Right.
releases free fatty acids locally that then can promote accumulation of lipids inside the muscle.
Okay.
And the subcellular stuff is all the lipids inside the muscle, where they're located, et cetera. So they're kind of separate, but they're also kind of related. In terms of the past work, we started looking at this in probably 2008, 2010, and been really looking at the subcellular lipid story for a while. And it's really hard. It's really difficult to take a frozen muscle biopsy and then differentially centrifuge it into these different fractions. Our first attempt, which is what you're kind of getting at, we published in 2012, I think, where we just sort of globally took muscle and separated it into all membrane compartments and then the cytosolic compartment just to see if there was something there. And it was one of these data sets that you probably get in your career maybe one time, where everything we looked at was significant. It was just remarkable. And it really let me realize that, okay, there's something here, that membrane lipids clearly are what are causing problems, whereas the cytosolic lipids, i.e. the lipid droplet, is not as much of a problem. So after we published that, we realized that's still super crude because you can't take all the membranes and just lump them all together. You're not going to learn anything there.
Right.
And so then that just sort of took us down the road to developing more precise ways to separate these lipids out of different membrane fractions. And that's ultimately what helped us with this paper. We had a previous paper before that was cross-sectional that compared people who were endurance trained athletes, lean individuals, individuals with obesity, and individuals with type 2 diabetes. So we had this nice kind of cross-section that spanned the physiological range of insulin sensitivity. And it also showed that these localized lipids were playing a role, but it's cross-sectional, right? And so we needed to really try to dive in and see what we can do to figure out what are the important localized lipids that are impacting insulin sensitivity. So we picked the hammer, which was this study, which is combined weight loss and exercise training. We figured that was a very powerful combination to make people more insulin sensitive. Let's take people through that who are already pretty insulin resistant, i.e. people with obesity and prediabetes, and see how things change. So that's kind of how everything led to this paper.
Yeah, so let's get to more about this paper. So you described a little bit, but maybe go into a little bit more detail about your subjects and the study protocol that you used for this particular paper.
Yeah, good question. This is sort of just a classic intervention study. So it was 12 weeks long. These were people with obesity, and most of them had prediabetes as well, because we were really trying to find people that were insulin resistant so we could have a big delta for insulin sensitivity change. Well, first we phenotypically tested them, right? We deep phenotyped everybody. They had insulin clamps with a muscle biopsy at rest, and also 1 hour into the insulin clamp. We had isotope tracers to quantify glucose turnover and uptake, DEXAs, the whole 9 yards in terms of phenotyping people. And then they went into this intervention, which was really difficult for the people because we cut them down to a really very low calorie diet using high protein, low calorie, kind of old school dietary interventions. Everybody had shakes, and we had 4 or 5 shakes per day for these individuals, and then they could have 4 or 5 servings of fruits or vegetables. And it ended up being around 1,000 calories a day. It was really, really low calories. And at the same time, we said, good luck. Now you get to also start exercise training. So people really, really struggled at first in terms of the ability to pull this off. So the exercise training was 4 times a week, supervised. Everybody had their own person that was helping them out. It was progressive. All the normal things that you think about with whole body aerobic activity. Most people were walking. Some people did, rowing and cycling. The smart people did cycling, but that's a whole different story. Most people were just, walking on a treadmill, and towards the end, they were starting to run, actually, which is really, really cool. And then the weight loss intervention, we had weekly sessions with people where they were becoming more aware of nutrition and the importance of nutrition and how to lose weight. It was a lot of just education component. And we tracked their weight every week, and it was a 12-week intervention. The important part is that after the end of the intervention, for 2 weeks, we weight stabilized people. And that weight stabilization, I think, is really important. I think it actually kept us from seeing a lot of differences, honestly. I worry that some of the people were eating too many calories, and we kind of obscured some of the differences that were there, but that's maybe a totally different story. But so we had 2 weeks of weight maintenance, and then they kept training during that period of time. And then before the post-intervention testing, where we repeated everything that they did beforehand, we had them rest for 2 days, so we weren't measuring the acute effects of activity. And then we had them weight stable, and they had a diet that we fed them for, I think it was 3 days. Well, no, it was a week, actually. It was a week before they did the final intervention. So we had dietary control before and after.
And they had a clamp and biopsy at the end as well.
Yeah, sorry,
right?
the whole thing. Yeah, so
Yeah.
clamp, biopsies, tracers, everything.
And so that was the big advance. The previous work you had done was cross-sectional, but here we have a repeated measures.
Exactly. And the whole idea, again, was just to kind of tunnel down into what are the important species of lipids in specific parts of the cell that could be related to the increase in insulin sensitivity, to give us some feeling for what's going on. Because there's so many species of lipids and all these places in the cell. It's just a really muddied waters right now. So we were trying to figure out what are the important ones we should need to focus on in terms of trying to develop some sort of therapeutic.
So one thing that occurs to me is if you're walking versus cycling or rowing, that you're getting additional load bearing in the muscle that you're pulling the biopsy from, right?
Right.
Were you able to pull out, you probably weren't powered for this, but were you able to appreciate any differences there in the IMAT? No. Okay.
No, didn't look at it. Yeah.
Okay. All right. Sorry, that was kind of in the weeds there, but
no. It's a good question, right? Yeah. We had everybody walk at an angle on the treadmill to try to recruit their vastus a little bit more, because this is the common complaint when people look at walking. Yeah.
Right. So your analysis really drilled down to the lipid species present and also the transcriptomic regulation in the muscle tissue of these volunteers. You even made the point that one of the most unexpected and exciting findings, the changes in nuclear lipids after combined weight loss and exercise training interventions, right? That you didn't expect those changes. Can you expand on that thought for us and what you think those nuclear lipids specifically may be contributing to the overall physiology that you observed?
Yeah, so this is where I'm gonna get a little woo-woo, because I don't know is the bottom line. But
that's all right. I set you up
for
important.
Yeah, and so in our cross-sectional studies, we actually found that nuclear lipids, when they were ceramides and triglycerides, were negatively related to insulin sensitivity.
Okay.
But the nuclear DAGs, whether they were a 1,2 DAG or 2,3 DAG, these isomers, were positively related to insulin sensitivity. And I thought that was a little crazy, because everybody thinks that DAGs are always bad. So in this intervention and in subsequent interventions that we haven't yet published, we're finding nuclear lipids changing dramatically in these interventions. And it's something that I've never really thought about, but it's really fascinating.
And these are specific, sorry to cut you off, but these are specifically the nuclei of the skeletal muscle, right? Not the adipocytes that may be embedded in the tissue.
Exactly, because I actually physically, I was physically the one at the dissecting scope pulling away the adipose tissue for the muscles, and we weren't just measuring contamination. And that's ironically how I got into IMAT, because I was physically pulling them away. I'm like, hey, people with diabetes have more of this stuff. This is weird. Anyway, so I think what's going on is that these DAGs in the nucleus could be regulating transcription factors and ultimately gene transcription. And this isn't as crazy as it might first sound, because there's other lipids that we know do this. Glycerol lipids, like phospholipids, do this. Long chain acyl-CoAs, leukotriene B4, polyunsaturated fatty acids. These are all known regulators of gene transcription. So it made me wonder, could DAG be a, up to now, unknown regulator of gene transcription that could be causing or influencing some of these changes in insulin sensitivity after these interventions? What was interesting with this is the nuclear DAGs weren't related to PKC activation, which is the sort of commonly accepted way people think DAGs impact insulin sensitivity. But it made us really think that maybe these are changing gene transcription, and we tried to pursue that a little bit. But ultimately, we have other studies going on where we're trying to do this a little bit better. Because I really think that these changes in nuclear lipids have been going on underneath the hood without us knowing it, because we've just been taking the whole muscle and looking at these changes, and it could be important. And for instance, there's been a couple papers that have been published that showed that after endurance training, DAGs increased in the whole muscle. Francesca Amati, now a collaborator, she's awesome, published this in 2011. And everybody was scratching their head. How can these athletes, after they become more trained, have an increase in DAGs in the whole muscle? And we didn't really understand it. I think it actually might be happening in the nucleus, because there's a ton of lipid in the nucleus, and it might be driving some of these endurance training adaptations.
And is there any evidence for the DAGs in the oxidative versus glycolytic fibers being different? Is there anything there?
It's a great question. We haven't been able to appreciate that because we take our muscle biopsy and we fractionate the whole thing.
Yeah.
We use 60 milligrams. It's a ton of muscle wet weight for this, and we haven't been able to tease apart fibers. I think people might be able to do this in an animal model where they have, more heavily distributed slow versus fast fibers, that kind of a thing. But we haven't been able to do that in humans based on the biopsies that we've been taking.
So you saw an increase in whole body insulin sensitivity with the combination of exercise and caloric restriction. But when you looked at the molecular level in the muscle, you didn't see those changes in insulin signaling. So what do you think's going on there?
I don't know. This is a good question. Either we're terrible at Westerns or there's something else going on, right? And it
No,
could be
I
both.
don't think
I don't
that's
know.
it.
I think the timing's okay. I mean, we followed Jeff Horowitz's paper from a while ago where he actually did repeated biopsies during a clamp and showed peak insulin action at an hour into a clamp. So that's when we did it. But this has actually been shown before. It's not super new or unusual.
Yeah.
And there's a couple explanations for it that people wave their hands around, and
Yeah.
I've now joined that chorus. But there is this thought that you don't need full PI3K activation in order to have full stimulation of AKT. You only need a little bit. So that's possibly one of the things that's going on. I also think that there could be changes in insulin signaling that are only happening in specific submembrane compartments that are important, like in the caveoli. We know this is an intense area of membrane signaling. We can't discriminate that. Maybe that's what's going on, whereas in all of the sarcolemma together, it doesn't really matter. And it could also just be AKT translocation of the sarcolemma that's different, even though it's activated. It needs to be working at the sarcolemma. So there's a lot of possible reasons. But when I back away even further, it seems more likely that it's not actually muscle insulin signaling, but it could be the microvasculature, because we know that the microvasculature also responds to insulin, and it also is insulin resistant. And could it be that we actually had an improvement in vasodilation with the insulin delivery that allowed more insulin and glucose delivery to the muscle, that allowed more glucose uptake by mass action and with glucose transporters, of course? So could it be something that we just missed entirely? And so I think the microvasculature is probably what I would guess is happening, but I have no idea is the answer.
Well, Bryan, are there any other key points or thoughts you'd like to leave us with as we close this interview?
I think I would like to just focus on IMAT a minute, just because I don't think enough people are thinking about it. The data we published a few years ago showed that IMAT is as bad as visceral adipose tissue in terms of its ability to cause insulin resistance, at least in vitro. We've subsequently published that IMAT's more inflammatory than visceral adipose tissue. And we just finished a study a couple months ago, and we have some of the prelim data coming out. And that data shows that IMAT is actually worse than visceral adipose tissue. And so I really think IMAT is a unique, untapped area of discovery for the field. And I just want to encourage more people to get into it, because I think there's a lot to learn there. And I think we have this depot constantly bathing muscle with factors that are causing insulin resistance, loss of muscle mass, loss of muscle strength. I think it explains the age-induced sarcopenia, obesity sarcopenia. I think it's a really, really important player that we don't know very much about it. And so I just want to encourage anybody to start looking at it. I'm happy to share everything we know because I think more people need to be studying this thing.
Awesome. Bryan, thank you so much for joining us today and sharing your expertise and discussing your recent work on IMAT and intramuscular biolipids in weight loss and exercise training. I encourage the listeners to read the full article in the December issue of Diabetes. Thanks again, Bryan.
Thank you.
Today, we will be discussing December's paper of the month titled Proteogenomic Discovery of Novel Open Reading Frames with HLA Immune Presentation on Human Beta Cells. We are joined by co-first authors Kathryn Walters, postdoctoral researcher in the Mukherjee lab at CU Anschutz, and Roberto Castro-Gutierrez, a postdoctoral researcher in the Tang lab at UCSF. We are also joined by co-senior authors Dr. Neelanjan Mukherjee, assistant professor in the Department of Biochemistry and Molecular Genetics at University of Colorado Anschutz, and Holger Russ, associate professor in the Department of Pharmacology and Therapeutics at the University of Florida. Welcome, and thanks to all of you for sharing your work with us today.
Thank you.
All right, as you guys may or may not know, we like to start with an icebreaker question. And since there are 4 of you, I'm going to try to keep it simple for me. I want to hear about your favorite science fact. All right, Roberto, start us off with your favorite science fact.
Okay. My favorite science fact and something that I've been really amused since I was doing my PhD in Holger's lab is that we can reprogram patient cells into stem cells and make any organ we want. That still like blows my mind. And I get really excited about cell replacement therapies. And then if someone is needing an organ, we have the ability to like actually make it for them. Of course, this is the early stages and everything, but it's something that gets me really excited.
That's a good one.
That is a good one. Neil?
Mine's like more of a observation of molecular biology actually happening in action. And it actually has to do with some of the techniques we used in this paper. So like the fact that we can actually see the ribosome and translation happening on any individual mRNA at any given time by doing a random set of, steps in a tube and then visualize that is really cool to me. And it's just awesome to see molecular biology in action.
Yeah, absolutely. Absolutely. Kathryn, can we come back to you?
Sure. I guess if we're taking it project orientated, I just think it's just super cool how we can take, all of these skills from different labs and bring them together. And it's, this is something that we didn't have the ability to do, 10, 15 years ago, projects like this, having us all on different coasts. And I think it's cool to see, kind of the future of science kind of coming together in a paper like this, talking about multiple omics, talking about different, computational approaches, different biological skills. We're writing the facts right now.
Yeah, that's great. All right, Holger.
So my favorite science fact is that we can recapitulate a lot of biology that happens in human now in a dish, right? And this has been the base of this paper, but then more pointed onto Roberto. So when we were together in the lab, I'm still marveling about this riddle that we have in the lab. If a PCR doesn't work, we have a PCR candle, and if you light it and run the PCR next to this candle, it suddenly works. And we're still trying to get to the bottom of why this is the case. But facts remain. We've done it many, many times, and it seems to work. So this is the next thing that we really want to get down to the bottom of the barrel to understand why this candle works.
I think we need a candle like that in our lab.
Yeah.
Can you
Maybe
sell this
a
candle?
glucose clamp candle.
Yep. Darlene, consider the ice broken.
Yes, definitely. Awesome. Well, I think like as evidenced by the fact that we have 4 of you here that we're talking to about this paper, it really highlights that science is a team project. But is there anybody else that participated in this project that Neil or Holger you would like to acknowledge?
I really want to thank many members in my lab and in Neil's lab, right, for the discussion and making the little things happen. Because as you probably hear later from us, there had to be a lot of little steps or techniques and improvements come together to really make this paper happen. We worked many years on this. And then I also want to acknowledge many researchers in the wider type 1 field that have encouraged specifically us, right? Like Roberto, when things look challenging, because as I said, it took us quite some time to actually get this project off the ground and working. And there I also want to state that this was really when Neil and I started to talk and brought our respective complementary expertise to get this going. So I also want to thank researchers in the field and highlight that they have encouraged us to do maybe break new ground in the proteogenomics area, right? Which opens, in my opinion, a whole field of study focused on immunogenicity of beta cells in the type 1 context. So yeah, all the individuals that made it happen.
Neil, anybody else that you would like to acknowledge?
Yeah, I just want to add a little more specifically, Maki Nakayama for working on some of the T cell response experiments, and Tim Fugman and Ernesto Nakayasu for the immunopeptidomic and proteomic expertise, respectively. As Holger said, yes, it required sort of our complementary expertise, but then all these other real experts in the field. I mean, none of these things are trivial, and putting it all together is what it really takes. And I agree with also the broader backdrop of what Holger mentioned
Yeah.
in both in many different research communities.
Awesome. The next thing I'd like to do is just have each of you, Neil and then Holger, kind of give a high level overview, quick snapshot of what your research program is about. And then maybe we can, after each of you do that, we can talk about how you all married your expertise to generate this project. So Neil, go ahead.
Sure. So our research is really kind of like basic science, foundational research, trying to understand how regulation at the level of RNA, messenger RNA, non-coding RNA, is crucial for, human disease. We particularly have focuses in understanding translation, so both like normal translation and translation in stress conditions with respect to different types of cancers, and then more recently, beta cells and diabetes through this collaboration with Roberto and Holger. So we're really interested in these sort of like RNA regulatory networks and kind of systems bio approaches to understand them.
Holger?
Yeah, so I have a long-standing interest in type 1 diabetes, and that's specifically in, how can we understand the disease, how it manifests, the how and what. Can we use this knowledge to prevent and treat, right, the disease? My lab and we have been working on using human model systems, stem cell technology to really reproducibly generate cells to study them afterwards in particular aspects of the disease. And this is how this whole project came about. And I won't say more, but we basically had the tools to manipulate human beta cells to a degree that allows us to look really at what is the translation happening. But then we're not experts in this, right? But then really Kathryn and Neil came in and really took this project and put it to the next level. So this has been a thrilling experience. Yeah.
Yeah. So that's basically how the project came about, Holger?
Yeah, in a sense.
Yeah, so Roberto gave a chalk talk
Exactly.
in our RNA Bioscience Initiative series. And like afterwards, it was just so obvious that we needed to work together on this. So I would let Roberto like kind of take that part over, and Kathryn had rotated
Yeah,
in Holger's lab, so there was
yeah,
already
that,
like a tight relationship. Like,
Yeah.
yeah.
So
Yeah,
maybe I
that's really
just
cool.
throw in there that, for us, that was ideal because there was this natural synergy, and then these two great individuals that really took the project and started running. And that's what you dream of, kind of what's happened. So
Yeah,
but
that's
they can
awesome.
tell us more
Yeah.
about it now, probably.
Yeah, I can say really quickly, but basically how these came to be was that I was studying molecular biology, so I was learning a lot about RNA biology and translation. I became really interested in it, and of course, always having in mind my home lab with diabetes, I was thinking how to merge these two things together. And I was more excited about the fact that in cancer, some novel open reading frames or some translation defects can lead to novel peptides being presented. And that blew my mind. I was like, we can definitely do that in the space of type 1 diabetes and autoimmunity. But like Holger has said, like we're not experts on this. So I was looking for someone to help me with the RNA and ribosome profiling side of things. And that's when we talked to Kathryn.
All right. So Kathryn, let's talk about the paper. All right. Your work used a system proteogenomic approach and identified novel or annotated open reading frames, or NUORFs. Is that how we're saying it?
Yeah, that's how we like to call them.
Okay, NUORFs in beta cell model systems. So can you tell us about the rationale for this project and why this was an important strategy to apply specifically to beta cells?
Yeah, so the real foundation of this approach, kind of like Roberto said, had been used in cancer research before, but ribosome profiling is just a super powerful method, and it's kind of the foundation here. It's giving us a chance to look at what is being translated in these cells. And beta cells themselves have such a unique and specific translatome. Traditionally we just tend to stick to like canonical peptides and products, and there's a whole world beyond that. We kind of merged our two labs' expertise, and I'll let Roberto kind of answer the model system that we used aspect of this, but the proteogenomics approach, this large data set is kind of like, let's gather as much information as we can and illuminate, right, the dark areas that we haven't been looking for, and let's look for these peptides that nobody else is looking for that could be causing problems. And being able to do that really came down to the model system that Roberto and Holger had developed, and was a super nice platform to have as well.
So before we get to the beta cells, maybe can you give us just a quick overview of the ribosome profiling, how it works, what, just the 30,000 foot for us?
Yeah. So ribosomes, right, are going to translate upon the mRNA and create that peptide. And ribosome profiling, essentially, we freeze the ribosome in place, and then we're going to sequence the little bit of RNA that ribosome is sitting on. And this is about 30 nucleotides, and it allows us to then align it back to the genome and see where on this mRNA that these ribosomes are sitting. And so we look not just for the canonical open reading frames, but we can look in the UTRs and maybe genes that are not typically thought of as protein coding. And so that kind of came up in this paper further on, where we discovered the open reading frame in the 5' UTR, the TYK2 gene. And so by looking at all of these options, we were able to come up with this list of 1,000 NUORFs that we think the field should pay attention to and watch for in their future research.
Yeah, thanks.
That's a great explanation. Roberto, so Kathryn already mentioned it. Can you talk a little bit about the model systems that you used for this paper? And then, keeping in mind that Kirk and I are in vivo physiologists.
I usually put dumb before that.
Yeah. So explain or touch on the strategies you used to identify those NUORFs.
Yeah, of course. So when we went to Neil's lab in order to collaborate with this project, they told us that they needed a large amount of human beta cells for this project in order to do the ribosome profiling. And that was a little concerning for us because human beta cells are very scarce. tissue, especially functional human beta cells. So, but our expertise is into generated stem cell derived human beta cells. So we accepted the task in order to generate a lot of these cells. However, the other problem with, or the other challenge with ribosome profiling was that we need a heterogeneous population, right? So that would represent another challenge for us because the cells that we generate, they are heterogeneous in which they contain other type of endocrine cell types. So in order to overcome this problem, what we did was to tag the ribosome only on the beta cells in order for us to be able to pull down ribosomes that are only expressed in beta cell. So in order to do this, we differentiated large amounts of stem cell derived beta cells and then transduced them with a lentivirus that has a ribosome protein tagged with FLAG under the control of the insulin promoter. So we did a lot of experiments to show that it was specific and then it works and that it's comparable to whole ribosome profiling. That's the methods that we use to try to pull down ribosome-associated RNA fragments from human beta cells.
Maybe I jump in here and say that Roberto didn't tell you about all the efforts where we tried genome engineering to stably integrate in the human pluripotent stem cell, like minimal promoters in so-called safe harbors. And we learned the hard way that a lot of them get silenced upon differentiation into definitive endoderm cells. So what he described is the polished version of a year-long process of finding out how we can achieve this to get beta cell specific pull down of ribosome mRNA complexes. But that's just me.
Are you being modest, Roberto?
Where's the trauma hiding the truth?
It's the PTSD.
That's right.
No one wants to listen about the negative data, I guess.
So the crux of this paper is really the NUORFs. And you guys have identified almost 1,000, is that right? NUORFs from these beta cells. And then you went on to characterize and understand the properties and potential biological roles of some of them. Can you tell us a little bit about what you found and why you think this was interesting? And I guess we'll go back to Kathryn for this question.
Yeah, so I think the number we landed on, right, was 986 NUORFs, if I recall. And we did some high-level characterization, kind of looking at different properties that they might have, such as length, or like how well they're being translated. And then we really went on, our list is actually much more expansive than that 986, but these 986 are kind of our high confidence targets that we'd really like the field to focus on. And they are ones that not only do we really believe have strong evidence at the ribosomal level, but we also found they had proteomic support. And then we also have some of them that have the beta cell specific as we denote them. And that's just meaning they haven't popped up in any other ribosomal profiling experiments from other cell types. And so these are ones for everybody to really kind of watch out for. And then we kind of pulled out one TIC2, which we found is actually fairly recent in evolutionary development. So it's primate specific. And this TIC2 gene actually has some SNPs associated with type 1 diabetes. And so these are kind of located near where this UOR for upstream open reading frame would be. And it's just kind of highlighting really the powerful approach that we have here in identifying these. And I think you could spend, a whole career really characterizing these NUORFs, and we've just started. And that's kind of why we want to get this paper out there as a resource so that others can really follow up on this work and continue, because they're very exciting, this list. I think we're very excited to share this with everyone.
Great. Roberto, can you follow up on that and talk about the link of some of these NUORFs to type 1 diabetes?
Yeah, gladly. Like Kathryn said, after getting all of these lists and potential great NUORFs, we divided for this paper 2 areas of investigation, which Kathryn just described, TIC2 part of the story. So for the type 1 part of the story, actually, And the part that I was more excited about is to understand the immune system crosstalk with the beta cells. Like we mentioned before, all of these nerves have the potential to be presented on HLA class 1 molecules on beta cells, and they can actually interact with the immune system. So as a proof of concept and try to see if these nerves actually can be presented, we perform immunopeptidomic analysis of these stem cell derived beta-like cells in order to curate the two lists that we will get, the list of the presented peptides by mass spectrometry, and then blast that list to the new list that Kathryn and Neil provided with us with the nerves and see if we can identify and we can see if these nerves are being presented to the immune system. So that is very important because in type 1 diabetes, we know that it's a T cell mediated disease, and these T cells come to the beta cells and destroy them. And something that still perplexes me in the field is that we have a ton of T cell receptor sequences that we know they're important and relevant for type 1 diabetes development, but we don't know what they're specific for. So these novel peptides, we wanted to know if they're presented on the HLA class 1, and then I can activate T cells. So we believe this is important because there are T cell receptors that have been identified that are relevant for type 1 diabetes that we still don't know what peptide they're recognizing. So by finding this new list of potentially presented newer peptides, then we can try to see if these T cells are specific for those novel peptides that no one has identified before. And the part that is exciting for us is that if this happens in the beta cell, but not in the thymus, that will actually give us more confidence that there's selection escape from the thymus of these autoreactive T cells. So basically can give us a new way to see if these T cell receptors are actually being activated by these nerves that no one has identified before.
That's great. So, Neil and Holger, are there any other key findings you'd like to mention, or maybe give us a little nugget of information of any future Mukherjee-Russ lab collaborations?
I'll say what I sort of see, and then I think it's actually helpful because we have very like complementary approaches and expertise. So I think you'll probably get multiple different answers from each of us. I think one of the things I'm really excited about is trying to understand the mechanisms that control the production and the presentation of these neurons in beta cells in the context of, stress responses that are associated with the sort of etiology of T1D. And there's a whole host of really interesting pathways that could be important and ultimately could be either targeted or manipulated to try to control this presentation. And I think the more sort of obvious clinical and translational part, I'll leave that part to Holger because I think he would really be more eloquent in describing that.
Thanks, Neil. So as Neil alluded, right, this opens a whole Pandora box, at least in my mind. First of all, how is the human beta cell responding to different stress conditions? And does that then ultimately contribute to an increase in immunogenicity, right? We have some data there, but we still don't understand why actually there's a break in tolerance. And now we have a list of molecules that actually could, and this is ongoing, can be matched with TCR information from T cells that are found in remaining islets of type 1 patients, right? To really get on, do we miss antigens that are critical for disease development? Are they shared among patients? Do we need to stratify better? I mean, this is just one aspect, right? As Neil really nicely said, can we understand the mechanism, how they're being produced? And then can we basically manipulate it? And what I maybe want to say in the end is like that, this is what in a way science is about, to not take what we know as granted, but actually think of where are the boundaries of what we can detect and what we know, and how can we push it there? Because, then others can start hopefully thinking of these aspects and integrate that into their research program, because it's not one lab, 5 labs. It really takes a village, right? And the hope is that it really triggers innovative thinking in many other labs that have the same goals than like us, like helping humankind, right? To treat patients, basically help parents with kids that are sick, to make it better. And this is what I think.
That's fantastic.
Yeah, I think that's great. Well, Kathryn, Roberto, Neil, and Holger, we want to thank all of you for joining us today, for sharing your expertise and for discussing your recent work on novel open reading frames on human beta cells. I encourage the listeners to read the full article in the December issue of Diabetes.
Welcome to another edition of Sweet Talk. I'm Kevin Williams, and I'm joined by one of our co-hosts today, Dr. Kirk Habegger. Today, we're focusing on how we assess metabolism and how new technologies and analytical approaches are changing what we can learn about energy and glucose balance. We have 3 experts to help guide us through this discussion. We have Dr. Alexander Banks, who is the director of the Energy Balance Core facility at the Beth Israel Deaconess Medical Center at Harvard Medical School. We have Dr. Corey Holman, who is the technical director of the Rodent Metabolic Phenotyping Core at UPenn. And we have Dr. Louise Lantier, who is the Metabolic Pathophysiology Core Managing Director in the MMPC at Vanderbilt University. Welcome, and thank you all for joining us today.
Thanks, Kevin.
Yeah, thanks for having us.
So before we get started, we'd like to give you all a little bit of an icebreaker question. I think we'll go with the outside of the lab. Are there any hobbies or interests you enjoy that might surprise some people? Corey, let's go with you first.
All right, thanks. Outside of the lab, I love camping and hiking, and I actually got married in May, and my husband and I are going on our honeymoon in New Zealand doing just that in December.
Oh, nice.
So very exciting.
Now that's the country of adventure sports. Do you have anything lined up along those lines?
We might do some whitewater rafting. We're Lord of the Rings nerds, so we're definitely going to Hobbiton and Mordor, which is Tongariro National Park. So we have some fun things planned.
Isn't this the home of the is it a Zorb? Is that right? The balls you crawl into and then
Zorbing.
Pull down the hill.
Yeah.
Zorbing. Okay.
Yeah.
I remember
you did see that too.
Yeah.
I did that when I was in New Zealand.
Would you recommend?
Well, there's 2 options. One of them, they can strap you into the inside of the Zorb, and the other one, you get into a pool of water as the ball runs down the hill. I did the strap-in version, and I'm pretty sure I had a concussion for a week.
Oh my goodness.
So to each their own, you know. Alex, what about you? Any hobbies or interests?
Yeah, I'm into fencing. I've been fencing since I was in high school, and I was quite competitive in college, and now I do it at a somewhat less intense level. Had a competition yesterday, and I just love it. There's so much technical aspect to it. There's a lot of strategy as well as physical, and basically any exercise that you like to do is probably a good thing. So, and I just love doing it. So I'm a fencer.
That's really cool.
Yeah, I think this is our first fencer.
Yeah, I think so. And Louise, what about yourself?
For me, it's more what I don't do, which is different. So, growing up working on muscle physiology, every meetings I'd go to and the people I'd hang out with were avid exercise, fitness, runners. They'd meet up everywhere. And I was kind of the outlier, never exercising. So I do like to hike, but it can't be like extreme. So I was always more of a give me a book, give me a good tree to sit under. And that's what I'll be doing.
Nice. A little exercise the mind there. Yeah.
All right. So let's start with how much the field has changed, and specifically how much has changed in how we assess metabolism between new tools, new analysis platforms, and better statistical practices. Feels like we're in a little bit different place than we were even 5 to 10 years ago. So I want to start with Alex, and you've been a strong advocate for improving how we analyze energy expenditure data. What do you think has been the biggest advances in how we measure and interpret metabolism in both animal and humans.
So this is a really interesting area, and I thought a fair amount about this. Over the past decade, the measurements of metabolic rate, especially in animals, have really gone from specialized, centralized cores to such as the mouse metabolic phenotyping centers that Dr. Lantier works at. But these devices are now everywhere, where you have indirect calorimeters at many, many institutions. And instead of just having a few well-trained individuals at these sort of specialized centers, you now have this democratization, and there's a lot more availability of these sorts of experiments, which is great because there are more experiments and there's more data out there. But it's not clear that everyone who's running these experiments has been trained appropriately in how to set up the experiments and how to analyze the experiments. And I think that's a pretty important gap. And I think what we realized was without clear guidelines that were established from the field, someone who was new to it or they got their first data set, they would kind of wing it. They'd kind of try to figure out how to analyze an experiment on their own. And some of our instincts were working against us. So in our recent paper that we just put out on the standards, we had this standards paper on indirect calorimetry. There was one table showing in a very specific, narrow example of activin receptor signaling, there are 16 experiments that use 8 different units and 8 different ways to analyze indirect calorimetry data, which is a mess. That's a disaster, right? I mean, if it's kind of the same experiment on a couple of different mouse models, it should probably be the same sort of analytical system. So to first get everyone to recognize we have a mess and what's the step forward. So if you look at the field from a decade ago, there was a really influential paper in Nature Methods from Matthias Chopp, John Speakman, and others describing the best way to analyze these experiments. And it was a really well done paper. They were saying that the way to analyze metabolic rates is to consider the body mass using this analysis of covariance. And we can talk more about that in a bit. But that paper, it hasn't really stuck in a way where not everyone is using those methods, and it's kind of becoming less relevant with time, even though I think it is correct. And I had this watershed moment where I was getting very frustrated understanding why more people weren't doing it as put out in this paper, this Chopp Speakman paper. And I have a friend who's an editor at a big fancy metabolism journal. And we were postdocs at the same time. And I was asking him why his journal didn't have uniform standards. He knew what to do, and I trusted him, and I knew he was trying to do it right. And his answer kind of surprised me. He said, it's not the job of the journals to establish the standards for the field. It's the job of the academics, of the academic community to come together and establish the standards that the journals will follow and the funding agencies will follow. And so he kind of threw it back to me, and he said, it's kind of your job to do this and not mine. And that shut me up pretty quick.
So you already mentioned it, but let's talk a little bit about ANCOVA and what we may view as the correct treatment of the data.
Yeah, it's a little complicated, so let's try to get into it. But the main issue is our instincts are working against us here. So if you load more protein on a Western blot, you divide by the total protein and you try to normalize for that. And as a scientist, you try to always correct for the amount of material you're using. And in mice, it's not quite the same thing. So if the problem occurs largely in studies of obesity, where when you have a more obese animal or person, you have more fat mass. And you might have the same amount of lean mass, that is the muscle mass and organ mass, or you might not. But so our instinct is to take the total mass from an obese animal and divide the metabolic rate by that total mass. And that's the instinct, and we think that's got to be the right way to go. But because muscle mass uses so much more energy than fat mass, when you divide by the total body weight of an obese animal, you're artificially giving yourself a lower metabolic rate because you're incorporating this lower energy demand from the fat mass into that equation. And so I'd say if there's one take-home message, it's don't divide your metabolic rates by total weight. And that is the mistake that I see more often than anything else. And the big message from this paper is not to divide by body weights. Instead, we use this method as outlined a decade ago of analysis of covariance. And that's where you take the metabolic rate and you take the body mass and use a linear regression to try to see whether these groups are different. And it's really not that bad. And it gives you a really nice answer. So that's the way that we're recommending in this paper. And Kirk, you're an author on the paper. Thank you for being part of that. And
Yeah, my pleasure.
And I think that's the way forward.
I mean, Alex is being, is minimizing maybe his impact in the field. So, the CalR tool, making the ANCOVA accessible, freely accessible to the community, has really brought or opened the door to allowing all grad students, postdoc labs, cores to implement this method of analysis, the ANCOVA, easily and quickly, which was also another big barrier, 10 or 15 years ago.
Absolutely. Those legacy systems and the analysis on the software was very challenging, and the integration at the CalR was a great introduction by Alex.
So we had one of these systems that would give you data that was automatically divided by body weight. And to be fair, I think 30 years ago when they came out, it wasn't clear how wrong that was and how misleading it was going to be. But as the field evolved, the software wasn't evolving. And so we wrote this program initially for our own use, where you would just load in your calorimetry data, it would undivide by body weight, give you the correct answer, do ANCOVA very quickly. And we were encouraged by people such as Louise to make this a freely available open access app that's just on a website that anyone can use. And so the idea is you don't really have to be an expert. You don't have to be the cognoscenti. You can just upload your data, visualize your experiment, get the correct analysis of covariance result, and you can be done in about 10 minutes. And I think that has helped a lot of people.
And I want to bring Corey into this discussion. Running a large phenotyping core, researchers are coming in and bringing you study designs on energy expenditure, calorimetry. What are some key strategies for collecting accurate calorimetry data that you see that making sure that the setup itself doesn't introduce an error?
Yeah, not only, Alex mentioned that many people, not just core facilities now, have these pieces of equipment. And, with these highly sensitive pieces of equipment, you can't just put a mouse in on a Monday, take it out on a Friday, and expect to have perfect data. You have to look at the data every single day. Are we getting body mass data? Are we getting accurate food intake data? Are mice taking food out of the food hopper? The machine's recording it as food intake, but caching the food on the bottom of the cage, that is it's not accurate data. So what we do in our core facility and what we advise people to do as well with their own machines is to, take a look at the data daily. If you find any food on the bottom of the cage that is erroneously counted as food intake, we measure that by hand. We then have to correct on the back end and remove that from the measured analysis. And it just takes a really careful hand to make sure this data is as accurate as possible.
I would also add that, calibrating and taking care of the equipment is not, a once in a blue moon, once a year, 2 hours type of job. You can get really good data out of a system if you take care of it really well.
I second that wholeheartedly. And we have two systems here at Penn. One our core facility runs, and one we train others in order to run. I can tell you the amount of things that break on our system versus the one that other people run. There's a significant difference. So although we train people as perfectly as possible, things are still, so involved with these calorimetry studies that I think one of the next advances in the field is to define real standards on how to run these studies. What is the best methodology? How much acclimation time do the mice actually need to not lose body mass in the cages because they're in a new environment? And so that's something that, I'm really excited to help Alex and the International Consensus Committee on this stuff going forward.
So I want to jump in and just talk about how do you know when your machine is giving you terrible data? Because this is something that we struggled with a lot initially. So we're getting measurements of mice every 2 and 3 minutes for about 5 days. And even if you have a group of basically genetically identical mice, once in a while you get one and you can plot the energy expenditure versus body mass, and it's usually pretty tight. And sometimes you get a mouse that looks like it's just off in its own world, right? Its metabolic rate is twice as high, where its food intake is 3 times higher than all the other mice, but it's gaining the same amount of weight and its body mass is comparable. And so I'd love to ask, these experts, I'd love to ask Corey and Louise how they would tackle something like that.
Yeah, it's looking at the data, it's having a lot of experience having looked at many data sets and then deciding, is this actually an outlier, running outlier tests. But when you're collecting the data, you know, on hand, if I see a mouse that's a lot higher or lower, I always go to the tubing. Is the tubing connected? Nope, usually isn't. And then you can fix that on the spot. But that's also a reason why you need to really look at the data daily so you make sure that you're collecting the best data possible.
Yeah, we have a whole pipeline in our MMPC. So we check the raw data on every run, the raw, and our techs do that every week. And so they know exactly what every channel is supposed to look like, from some of the more obvious food intake and water intake to some of the more time it takes to communicate between the devices type of data streams. So we can pick up some things that are starting to go a little wonky very early on. And then, as Corey said, it's really looking at the data altogether. Is the mouse, does it have a higher energy expenditure, but also is its body weight higher? And is it also eating more, or is it also moving more? Does it make sense? Is there a clear picture of this mouse? Data makes sense based on its behavior and what it was doing in the cage? Or is there really just one piece of data that's really weird and everything else looks like the rest of the group? And that's okay, something might be wrong technically there.
An approach that we've gotten into recently is when you take the body mass of the mouse before and after the experiment, and you can see how much weight it's either gained or lost, and you try to correlate that to the changes in energy balance. So is the mouse eating more than it's burning, or is it burning more than it's eating? And you can have a prediction of how much weight it should have gained or should have lost if all of your calculations are correct. Or your food intake measurement is accurate, your energy expenditure is accurate. It should give you a pretty good prediction of, is this mouse gaining weight or losing weight? And when those values don't add up, it makes us seriously question, dig into the data. Was there food caching? Was there some tubing loose? But it gives us a first pass of whether the data is of a high quality if everything adds up.
Yeah, absolutely. That's 100%.
So I guess I kind of want to move on to, we've talked about a lot about the data here, and it's a lot of data that you get out of these machines, right? But then we're also collecting lots of other endpoints on the same animals. Some of it is molecular, some physiological, some behavioral. How do we make sense of all of this complexity? And what's the key to integrating all of these data types in a meaningful way? And I guess this is, to the committee here.
It's an unanswered question, right? So you get all of this different data, and I think we're getting more sophisticated with, say, feeding pattern analysis and the components of energy expenditure. But I think there are other things, pieces of metadata that we're not collecting well enough. So are you fasting a mouse for 2 hours or 4 hours? Sometimes you just say fasted. Is it an overnight fast, which is going to drive the mice into torpor? And you're going to see dramatically different lipid profiles and cholesterol levels and glucose levels, depending on so much of this other stuff. And to integrate all of these data sets together, you really need an accurate accounting so you can compare experiments done in your lab with experiments done in my lab. And I think that's sort of the missing piece right now, because without that, we won't really be able to replicate experiments particularly well.
Yeah, I think it's important to not only do the indirect calorimetry experiments correctly, but to also integrate it with, fecal calorimetry. So taking the caloric content of the feces after the run, because energy in and energy out. You can also do, clamp studies. Vanderbilt has been the pioneer in this method. Anything to do with insulin sensitivity as a metabolic phenotyping core. We advise people on, what to do and when to do it, and really to look at a whole picture of a particular mouse model. You need to know a lot of the different parts of the metabolism. That's getting their background information from the investigator, what they've already done, so we can advise them what to do going forward.
I love what I hear in that discussion of advising the investigators about their experimental design and their plan. I mean, you guys, I'm sure, have plenty of investigators that are coming to you on a regular basis, performing these experiments over and over with different experimental models. But undoubtedly, you have people that are completely naive and new to this type of experiment and metabolic assessment, and having somebody there with the expertise to help guide the setup of an experiment and decide potentially what could best answer the question at hand, I think is really valuable. And so I think one thing for the listeners is reach out to your core facilities and talk to these experts. They're there to help you and guide you through these sometimes very challenging experiments. And to follow up on this, I wanted to ask in regard to the consistency, the reproducibility, the translatable aspect of the data that we've all seen cases where phenotypes are not replicated across studies, they're not replicated across institutions. What can we do to make metabolic phenotyping more reproducible when labs are using different equipment, different systems, or different types of conditions? We've kind of touched on this a little bit as it relates to the standardization of ANCOVA or maybe using CalR as an analysis tool. What other things might be there for consideration? Corey, we can start off with you.
First thing is definitely talk to your core facilities, as you mentioned. I can advise someone all day not to divide by body mass, but if they take their data, run away, publish it without our knowledge, and don't acknowledge the core facility, that's a real problem. We love to help write methods. I love to help look over final figures before publication, but we really need the investigators to be accountable for that as well, and actually take the advice of your core facilities. So that's number one. And then Alex can probably speak more on this, but as part of the CalR, and he's writing a grant in order to create a repository where we can actually collect, just like, genomics. I used to be in single cell. We have so many single cell data repositories that are searchable. We really need this to define standards, to have people upload their metabolic data to CalR, and then it to be a searchable feature that we can go through. And Alex, feel free to take it from there.
Yeah, to the question of how do we make the data more replicable across sites is one that terrifies me, frankly, Kevin. I don't know if we run experiments at 10 different sites, if we're going to get the same answer 10 different times. And we had a small hint into this. The International Knockout Mouse Phenotyping Consortium was this incredibly ambitious plan to phenotype 3,000 different knockouts across 11 different sites. They did a whole bunch of different phenotyping, including indirect calorimetry. And I think indirect calorimetry was one of the most problematic data sets that they had, probably because of how they set up their experiments. It was very short. Maybe the mice weren't acclimated. But what they found was, of the knockouts that they sent to multiple sites, rarely did you get the same phenotype. And that was really a wake-up call. And looking deeply into their methods, they didn't really control for temperature that well. And multiple people have shown, including Mark Reitman and others, that body temperature is a huge factor in the metabolic rate of mice. And having mice at even 2 or 3 degrees difference in ambient housing temperature can dramatically affect their metabolic rates. And so a phenotype may be different if you have mice at just a couple degrees different. And so you might think your mice are at room temperature, but room temperature in Dallas might be different from room temperature in Boston. And because of that, you have these different things. So what I think we can do to really maximize the replicability is just record body temperature and record body weight and record as much metadata as we can. And as Corey said, we're putting together this, from CalR, we now have the Cal repository. And the idea is there will be another tab on the CalR page where once you've already uploaded your data and you've gotten your analysis, if you wish to voluntarily share your data, you can log in and deposit it. And then we're going to be able to see your phenotypes and my phenotypes and put them all together and look at them in some big compilation. And I'm really hoping that we don't get different answers depending on the different sites. I believe that all of these, the equipment is very comparable across sites. I think we're still just measuring grams of food and oxygen and carbon dioxide, which are the same wherever you are. And I think the machines are probably equivalent over long-term steady state experiments. I'm not 100% sure on that, but I'm really hoping that when we put it all together, we get some good interpretable answers.
That's a really good thing to be pursuing on the biorepository. So it'd be very interesting to see what comes out of that and see if we can get some standardization. And lastly, with Louise, Corey already mentioned it a little bit with the courses that are offered by the MMPC at Vanderbilt. And I think you're in your 17th year of maybe not you particularly, but the MMPC is in its 17th year of the isotope tracer in metabolic research, and you're taking some time away from that this week because it is going on this week, I believe, to join the podcast. So maybe you can talk a little bit about what's going on at the MMPC with the courses and trying to educate the field on these different methodologies.
Yeah, so the courses put together by the MMPC are really aiming at improving rigor and reproducibility by really being transparent about our methods, but also recognizing the fact that, as Alex was saying, for energy expenditure, there is a democratization of metabolic flux studies. There are labs trying to get into the metabolic flux field or using stable isotopes and really getting those types of measurements going. So yeah, this year we have, or this week, it's the 17th isotope course here at Vanderbilt. That's actually where I met Corey the first time when she attended this course.
Yeah, it's fantastic.
So
Please take it.
Yeah, it's a really intense course. It's a full 5 days, but you're going to learn everything you ever wanted to know and some of the stuff you didn't want to know about stable isotopes or radioactive tracers for use in cells, animals, humans, any type of model that you're looking for. So it's a really great course. And then we also have the hands-on glucose clamping the conscious mouse course that's also held annually. That was about a month ago at Vanderbilt, and that one is a lot more how do I clamp a mouse? How do I put catheters in a mouse? Hands-on, hours of one-on-one time with surgeons and clampers, and it's a really excellent course as well.
And can I shout out to your standards paper about how to standardize the clamping experiments? So the MMPCs had this paper that came out. I can't even say how many years ago it was, but what they were trying to do was get all of these expert centers together, compare notes, and issue standardized guidelines on how to perform these sorts of experiments. And this was very much an inspiration for our current efforts in indirect calorimetry, is to kind of get the experts in a room, have a conversation, work out the hard issues, and then put it out there for the community to read and comment on. So thank you for that.
Yeah, and that's really a major part of the mission of the MMPC as well, is to validate techniques and create standards. And when things are not standard, at least be open about it. Why did you choose 12 hours of fasting? Or at least make sure that your paper has that in there. Where do you take the blood? How often? Those types of little details that, for a period of time, the papers tended to want the methods to be reduced and shortened. And I'm hoping that we're getting away from that, and that now it's understood that, we want details in the methods.
Yeah, having taken both of those courses, we use your clamp methods here at Penn. It's just fantastic, and you guys have done so much for the field. And when I talk to my fantastic research specialists, I say we want to be just like Vanderbilt. So you guys are really who we look up to, both of your core facilities.
Yeah, that's awesome, because we love those success stories when folks attend these courses and then are able to translate that into their lab, into their home institution, and set it up. That's amazing. That's what we're striving for.
Not to add on, but I have attended those classes, and so has my staff, and that's exactly how we built our program as well. So they work. Now that's looking backwards, but I want to shift completely and look forward. Right. There's also a recently announced initiative from NIH to put more emphasis on human-based research, with fewer awards going to animal work. How do we think, as a committee here, this approach is going to shift experimental design? Let's start with Louise.
Yeah, so this is something that, as a consortium, the MMPC is spending a lot of time discussing and thinking about. There are definitely some initiatives being taken to validate these alternative methods, right? Whether they're organoids or cells, or maybe humanized mouse. It's still in vivo, but it's maybe more relevant to human disease. And how do we validate that? And how do we advise our users, our investigators that are going to come to us and ask us how to answer specific questions, which model would be the best? And one thing that's very clear is that, we're not here to push people to study mice. We're not, the reason we all got into this job is not to cure diabetes in mice. So, and if there are ways to do some experiments in cells or organoids or in silico or using genomics data banks, we're going to want to do that. So we're setting that up, and we're making sure that those resources are available to the users. But that being said, there are some things that you just cannot do in a human and cannot do in a cell and cannot do in a model system, and you have to do it in an intact mammal. Energy balance is one of those where I really struggle right now to envision a model that would not be a live mammal.
I have nothing to add to that. I think that was a phenomenal answer, Louise. If I knew how to model feeding behaviors, adaptive thermogenesis, the changes that happen with weight gain and weight loss, there's just so much that we don't understand about mammalian metabolism that we're still working really hard and we're making good progress on in mice, but is informing our approaches to human weight loss and weight gain. I don't see a way forward to do that in cells. Even human adipocytes and human hepatocytes in a dish together don't work the same way as they do in vivo. So I think we're going to be doing some mouse work for a little ways to go.
Maybe I'll before we go to Corey, I'll shift the question, right, away from what won't work to what will. What's some low-hanging fruit that maybe we can target to address this new initiative?
Yeah, so, well, we have a preprint that just came out. One of our collaborators, Daniel Wieland, a postdoc in Josh Rabinowitz's lab, he has an interactive web app called Explore Competitive Catabolism, which is a way to put in, does your mouse have insulin resistance? What is the energy expenditure? And it basically predicts what a glucose clamp would look like, what a metabolic cage study would look like. I think that's a fantastic start and a way to develop the best questions you want to actually study in the mice as a way to reduce the amount of mice you might need to use, the amount of times you might need to do the experiment. So going forward in the field, you can potentially start with that. But I think at the end of the day, if you give a reviewer a, quote unquote, model derived clamp, they're going to say, where's the actual clamp? And so we can never get away from mouse work. We all love mouse work, love working with rodents. But yeah, in order to reduce the numbers, we have ways to do that.
Yeah, very well put. And we didn't talk beforehand, by the way. That wasn't a planted question.
So it sounds like AI and machine learning have a little ways to go before they can replicate the mammalian studies. So that's good to know. I think this is a really great discussion. I really appreciate all of our guests for joining us, Dr. Banks, Dr. Holman, and Dr. Lantier, for sharing their insights and helping us see where the field is and where it is headed. Before we go, though, could you each let our listeners know where they can follow your work, contact your core facilities? Do you have a website, a lab page, or can the listeners follow you on social media? Louise, might start off with you with the MMPC.
Yeah, so the MMPC is a consortium. So we have a consortium-wide webpage, mmpc.org. The Vanderbilt MMPC, like all the MMPCs, has its own website. It's vmmPC.org. Our email is really easy. It's mmpc@vanderbilt.edu, and we're always listening. We're always there. And like Corey mentioned, it's important to talk to us. If you have questions, we're so happy to talk. And really, we want to talk before you do the experiment. That's really the best, where you're going to get the best bang for your buck, is if you talk to the core facilities before you do the experiment.
And Corey?
Absolutely. Talk to us. Can't stress that enough. Thanks, Louise. Our website is med.penn.edu/Rodent Metabolic Phenotyping Core. Please, my email is on that website. Send an email, reach out. Happy to connect on LinkedIn as well. We want to hear from you, and we want to help the field move forward as much as we can.
And Alex?
So the Energy Balance Core has an email, energybalancecore@bidmc.harvard.edu. But really, most of our work could be found at calarapp.org, which is where you find the free online web app CalR, which will analyze indirect calorimetry from any of the major manufacturers. And because of this, because people are analyzing really thousands of experiments every month, we get a lot of calorimetry questions. I feel like we're the world's calorimetry help desk. And so we get a lot of random questions. Some of them, are like, why isn't my file loading? And some of them are really deep, philosophical questions about how you think about these experiments. We're happy to answer them. So please feel free to reach out.
That's great. So listeners, please reach out to the panelists here. If you have questions, definitely reach out to your own core facilities at your own institution. They're there to help you. And as always, don't forget to like, follow, and subscribe for more diabetes bio content.
Thanks to all of you for joining us on the Diabetes Bio podcast. We hope that you're enjoying our discussion of the work that the Diabetes Journal is publishing each month. The feature articles we discuss in each episode are freely accessible at diabetesjournal.org/diabetes and are also listed in the episode notes for your reference. As with any podcast, it really helps us if you like, rate, follow, and subscribe to the podcast. And if you have any feedback for us, we'd love to hear that as well. We look forward to presenting to you each month.